world has now entered an aging society, and various diseases are also becoming more prevalent among younger people. The current drugs are difficult to meet the needs of diverse diseases. To significantly shorten the time required for drug development, it is necessary to provide diverse drug types and processes. National Sun Yat-sen University has established the Innovation Center for Drug Development and Optimization to conduct interdisciplinary integration. This center brings together biologists, pharmacists, chemists, and clinical physicians, more than 30 experts and scholars. Our center covers 4 major technologies in the drug development pipeline, including target discovery, compound design and screening, lead drug optimization, and efficacy verification. Our center provides new therapeutic targets for different diseases, various drug types, and efficacy verification in different disease models. Drug development is a lengthy process, primarily due to being the difficulty in target selection. The current number of known human genes is about 20,000 to 25,000. Additionally, the discovery of non-traditional genes such as microRNAs in recent years has made target selection of new drug developments extremely challenging. To overcome this challenge, our center uses multi-omics approaches, including genomics, transcriptomics, and proteomics data for precise statistical analysis to identify representative biomarkers. These are followed by the evaluation of functionality, clinical relevance, and drug feasibility, ultimately narrowing down from over 20,000 genes to a few hundred or tens of high-potential drug targets. This significantly reduces the scope of targets for further validation and development by the research teams. Moreover, the center has many experienced research teams that have been deeply involved in the field of drug development for many years. These teams, using both literature and their research, have identified many highly potential drug targets, laying a solid research foundation for our center. The construction and screening of new drugs include natural products and drug simulation and design. Natural products are the mother of modern medicines and the main source of new drugs. Examples include paclitaxel isolated from the Taxus brevifolia and the artemisinin derivatives extracted from Artemisia annua, both highly representative natural drugs. The sources of natural drug development range from traditional herbal medicine to microorganisms discovered since the 1940s, the marine organisms following the development of diving equipment. The research on marine natural products is one of the center's key research features. The separation of natural products mainly utilizes chromatographic methods. We use various silica gels or molecular sieves to separate different compounds, ultimately obtaining pure compounds through high-performance liquid chromatography. Once pure compounds are obtained, nuclear magnetic resonance spectroscopy, mass spectrometry, and circular dichroism spectroscopy are used to identify compound structures. Once the structure is confirmed, drug activity screening can be conducted. To date, we have purified over 500 natural products that can serve as targets for activity screening. Currently, among the top 200 drugs by global sales, at least 2/3 are chemically synthesized drugs. The synthesis of drugs includes the development of processes for macromolecular and small molecule drugs. Our center has rich experience in the development of antibody drugs, peptide drugs, and targeted protein degradation technologies. We also develop various new synthetic methods coupled with computer-aided drug design strategies to construct multiple important potential drug molecular structures. These new methods include using visible light and clean electricity as chemical energy sources and flow chemistry, a safe and reliable technique for large-scale molecular preparation. The center is also equipped with various analytical instruments such as high-performance liquid chromatography, often referred to as the chemist's eyes, to identify the purity of molecular drugs. Additionally, we have nuclear magnetic resonance and gas chromatography mass spectrometry to identify the composition of synthesized compounds. Ultimately, we hope to propose extremely simple solutions to truly difficult problems. Efficacy verification mainly includes 4 parts: cell testing, animal testing, pharmacokinetics research, and clinical validation. In the in vitro cell testing stage, we use the SELECT method to screen and optimize effective anti-cancer, anti-inflammatory, antiviral, hepatoprotective, and neuroprotective drugs, further optimizing their molecular structures. We then select suitable disease animal models for in vivo testing. Using advanced cryoscanning electron microscopy, confocal 2-photon microscopy, IVIS in vivo imaging systems, and flow cytometry, we can more accurately verify pharmacological effects. Currently, we have established various animal models, including cancer models for liver cancer, lung cancer, pancreatic cancer, and colorectal cancer. Chronic disease models include cardiovascular disease and diabetes, as well as infectious disease models such as dengue fever, enterovirus, and influenza virus. Effective targets can undergo further pharmacokinetic, toxicological, and safety assessments at our center. Once the targeted drugs have passed the aforementioned series of preclinical tests, they can be ready to apply for entry into the clinical trial phase. Our center has multiple clinical research physicians and has formed alliances with more than 10 hospitals in the greater Kaohsiung area, enabling us to obtain sufficient clinical samples for conducting clinical trials. This allows us to realize a fully integrated translational medicine center from laboratory drug testing to clinical application. Our center has already developed numerous lead compounds for different diseases, including small molecules and macromolecules such as peptides, nucleic acids, and other biologics. We hope that in the future we can collaborate with the industry to further develop these drugs, meeting market demands and promoting the industrialization of university research outcomes. The center also welcomes collaboration at all stages, in addition to providing target identification and drug development. If companies have lead compounds, they can entrust us with drug optimization, design, and efficacy validation. These collaborations could accelerate new drug development, bringing substantial benefits and breakthroughs to the medical field, while also improving public health and well-being.